The human genome contains a whopping 3 billion base pairs, or the building blocks of DNA. A genome refers to the complete set of genetic material present in an individual organism. Genome sequencing has become fast and affordable and is one example where huge biology data is generated. Biology now competes with astronomy, YouTube, and Instagram in terms of the volume of data generated. Availability of large data sets has lent a new dimension to the modeling of biological systems at all scales, from modeling of molecular phenomena to getting insights from patient data. An integrative approach that combines big data analysis techniques and mathematical modeling has brought in a fresh arsenal of tools for identifying patterns, getting correlations, and obtaining insights. In the physical biology laboratory, Professor Ranjith, Padinhateeri and team study the physical properties of DNA and chromatin using computer simulations. The human DNA is like a 2-meter-long twisted ribbon and fits inside the nucleus that is a few micrometers in size. It is like folding a 30-kilometer cable to fit inside a tennis ball. Inside the nucleus, sections of the DNA ribbon are wound on several ball-like protein structures to form nucleosomes. This collective assembly of DNA and protein is called chromatin.
There are two puzzles associated with chromatin. How the cells package this DNA inside this cell nucleus is a puzzle, and all our cells have exactly the same DNA. But our skin cells function very differently from our brain cells. So how these cells function very differently even though they have the same genetic code is another puzzle.
Epigenetics is a field that studies how the extra layer of information on top of the genetic code affects the gene expression and cell function. Professor Padinhateeri uses computational modeling and simulations to understand the chromatin structure and dynamics with the help of epigenomic data.
We simulated how these proteins are organized along the DNA, so we could predict the polymer property of this chromatin, how stretchable it is, how bendable it is, and so on and so forth.
What researchers know until now is that the genetic code required to make proteins is encoded in different regions of the DNA. Only the regions of chromatin relevant to that cell type open up to make necessary proteins.
Our simulations predict how the DNA folding dynamically change in the cells, allowing the cells to open up certain regions or close certain regions. What is amazing about these systems is that they are self-assembled, and these dynamic structures do certain function. So how they function at the right time at the right place is an interesting question that we all still study.
Professor Sanjeeva Srivastava and his team at the Proteomics laboratory use proteomics, or the large-scale study of proteins, to detect and characterize cancer and infectious diseases. Just like the genome is a complete set of genes, proteome is the complete set of proteins present in a cell, tissue, or organism.
Our ability to understand thousands of proteins in a very short time gives us a new way of looking at the disease biology. My own lab has utilized these technologies to understand the complex disease like brain tumors. What we realized that even the normal protein expression of the entire brain is not available. So if we want to compare what is the difference between the normal brain proteins and the diseased gliomas or any other type of brain tumors, the protein expression profiles cannot be compared. So first we tried to comprehensively make the brain proteome map, which was the normal protein expression of a normal brain.
Professor Srivastava's team created an open access database called BrainProt that has the whole proteome map of a normal brain. Later, they added the proteome information of brain tumors and other brain diseases. Glioma is a very aggressive type of brain tumor, which is often detected at a very late stage, because of which the life expectancy is low. Professor Srivastava's team is using proteomics to detect glioma early.
We utilized a proteome array, human proteome chip, which is composed of all the human proteins. We took almost 200 glioma patients, which is of different grades of glioma, and aim was to look at, are there autoantibodies produced in this blood which can bind to the antigens on the chip, and can we have a signature which can be utilized for early diagnosis of disease. So after doing the comprehensive work and then lot of data analysis and the machine learning, we could actually come up with a panel of the 4 protein markers which are indicative of early disease diagnosis of the brain tumor. And now we are working on commercialization of this along with the industry partners.
In the Glycobiology laboratory, Professor Balaji and team are interested in investigating the proteins involved in the synthesis and degradation of monosaccharides, the building blocks of carbohydrates. Each living being seems to use a different set of monosaccharides to make its carbohydrates. This is in stark contrast to the case of proteins and DNA. The set of building blocks in DNA and proteins of all organisms is pretty much fixed. Professor Balaji and team hypothesized that there is no fixed set of monosaccharides that is common to all organisms. There are more than 100 monosaccharides synthesized by living organisms, out of which information for biosynthesis of 55 monosaccharides is available.
We gathered information about the biosynthesis of 55 different monosaccharides. We looked at the pathways for the biosynthesis of these 55 different monosaccharides, and we asked the question, can we identify proteins which perform different steps of the biosynthetic pathways?
They developed a functional annotation pipeline, a computational method to scan whole genome sequences, and based on the specific knowledge of synthesis of monosaccharides, assign functions to those sequences.
We wanted to know if we can use this function annotation approach to find evidence to support this hypothesis. We scanned the genomes of bacteria for which complete genome sequence is available. We did about 13,000 different bacterial genomes we looked at, and what we found is that out of the 55 different monosaccharides, only 12 are most commonly found. The others are used by very small number of bacteria.
Professor Balaji found a huge variation in the monosaccharides produced by different strains of the same species of bacteria. This finding is of interest because the variation can be used to differentiate strains and design strain-specific drugs and vaccines. Professor Deepak Agarwal develops personalized computational tools and healthcare technologies by combining data science and mathematical physiology to improve disease management and critical care in intensive care units. Patients with severe difficulty in breathing are administered a mechanical ventilator to help them breathe. A mechanical ventilator is like bellows. It pumps air in and out of the lungs. If the timing, pressure, and volume of the air pumped by the ventilator do not match the patient's demand for air, it results in an abnormal breath that can injure the lungs. Professor Agarwal built a framework that can continuously identify and quantify the severity of some of the abnormal breaths.
A trained or experienced clinician can know the state of the patient by examining the waveform that are produced during the mechanical ventilation.
Professor Agarwal is trying to mathematize the knowledge that the clinicians have so that specific features in the waveform can be used to understand the patient state. He identified clinically relevant features of the ventilator waveforms and built a mathematical formulation to represent the waveform signals of individual breaths.
There may be 10 or 15 different kinds of breath a patient might be happening during their stay. We estimated around 100,000 breaths from total 13 patients. We came up with a very simplified framework, a very simple formulation to phenotype a breath into 4 different categories, starting with normal, mild, intermediate, and severe.
Knowing the severity of the breaths can equip the nursing staff to determine when the patient's condition is not good and to intervene appropriately to stabilize the patient.
One of my long-term goals is to build a platform that will enable data-guided optimal ventilator management.
Researchers at the Indian Institute of Technology, Bombay, are using big data analysis in interesting ways to unravel the secrets related to the DNA compaction, protein expression in diseases such as cancer, synthesis of carbohydrates, and patient care in ICUs.